Dr. Lisa Newman from York-Presbyterian Weill Cornell Medical Center will be doing a virtual discussion. Lisa, welcome.
Hello, it's an honor to join you virtually today for General Session 4 of the 2021 San Antonio Breast Cancer Symposium. I had the pleasure of reviewing the brilliant work of Drs. Barrio, Kilwazi, and Gerber, and I congratulate them on their powerful presentations. Dr. Barrio demonstrated that Black breast cancer patients experience higher rates of lymphedema following axillary lymph node dissection compared to white patients based upon prospectively acquired data from a rigorously designed screening study. Dr. Kilwazi presented race-related subset analyses from the I-SPY 2 trial demonstrating comparable response rates to neoadjuvant treatment in Black and white patients within the strata of distinct tumor subtypes. And Dr. Gerber demonstrated that sentinel lymph node biopsy indeed has fewer postoperative adverse patient-reported outcomes compared to axillary lymph node dissection, but it indeed features several unfortunate sequelae despite its minimal invasiveness. These three abstracts all contribute importantly to the conversation regarding disparities and health equity, albeit in different and to some extent tangential ways. Most of my breast cancer research career has focused on the study of race-related variation in breast tumor biology, and whenever I give a presentation on disparities, I always display these three survival rates of 60%, 43%, and 20%. These survival rates are completely unrelated to cancer outcomes. They represent the survival rates for the first class, second class, and third class cabin passengers of the Titanic. They also serve as a tragic reminder that achieving health equity for diverse population subsets must always begin with an analysis of the problem through the lens of access to care. Just as the third-class cabin passengers of the Titanic did not have equitable access to the lifeboats, it remains true today that communities of color in the United States, such as African Americans, are overrepresented among the impoverished and the uninsured, resulting in socioeconomic disadvantages that create formidable barriers to healthcare access, including cancer screening and treatment. Dr. Barrio eloquently demonstrated higher rates of lymphedema among Black compared to white breast cancer patients, and so we should also look at access to the various strategies that are offered to women in an effort to proactively address, prevent, and treat lymphedema. Services that should be routine, such as PT/OT and exercise programs, as well as more cutting-edge or even investigational approaches, including pneumatic compression pumps or surgical lymphatic restoration procedures, are all associated with cost and time commitments that may be prohibitive among patients with financial constraints or limited workplace flexibility. Dr. Gerber showed us that while sentinel lymph node biopsy is an improvement over axillary lymph node dissection with regard to postoperative upper extremity limitation in range of motion, it's certainly not totally free of morbidity compared to women that are able to avoid axillary surgery completely. And so, as we seek to de-escalate risk stratifying axillary breast cancer surgery by relying more upon genomics and primary tumor features to define systemic therapy needs, it's appropriate to pose the question of whether we can mitigate some of the disparities in lymphedema risk that Dr. Barrio presented through the widespread application of advances in de-escalated surgery for breast cancer. Well, this approach might be successful, but only if all components of advances in cancer care are equitably delivered across all population subsets. Unfortunately, however, historically this has not been the case, and lymphatic mapping sentinel lymph node biopsy is just one example where we have numerous studies documenting lower rates of sentinel lymph node biopsy among clinically node-negative Black compared to white patients. Some of these studies represent the earliest years of adopting the sentinel lymph node technology based upon SEER data, as shown here. Other studies based upon the National Cancer Database, as shown on this slide. And still other studies published as recently as 2014 by Black et al., utilizing the linked SEER Medicare registry, continuing to demonstrate underutilization of sentinel lymph node biopsy for Black compared to white breast cancer patients. And so if de-escalating axillary surgery is to be adopted as an appropriate strategy for decreasing lymphedema risk in African American women, we must be absolutely sure that tumor genomic studies are being equitably applied to the cancers of Black women so that we avoid understaging and undertreating African American breast cancer patients. Now, in addition to addressing disparities from the perspective of access to clinical care, we must also remember to prioritize access to research. Dr. Barrio speculated that their observed differences in lymphedema incidence for Black compared to white women could be due to race-related variation in inflammatory response. This is an intriguing and provocative concept, but definitely worthy of further exploration because we indeed see evidence of race-related variation in inflammatory and immune response in the context of both benign and malignant conditions such as keloid formation and multiple myeloma. In this presentation, Dr. Barrio focused on the AL&D cohort. It will be interesting to see if surgical toxicity disparities persist for the subset of patients that underwent sentinel lymph node biopsy alone. Of note, the study here by Black et al. Representing DSEER data indeed demonstrated higher rates of lymphedema for Black compared to white women following axillary lymph node dissection, as well as following sentinel lymph node biopsy. Unfortunately, however, we are behind the proverbial 8-ball in research studying cancer genetics and genomics with regard to diverse populations, and as shown by these pie diagrams representing genome-wide association studies, fewer than 3% of contributing individuals have African ancestry. Many investigators are now racing to fill this vacuum of missing information. The I-SPY 2 investigators, for example, evaluated variations in immune signature expression across racial groups, identifying interesting differences between Black and white women with hormone receptor positive, HER2 negative tumors. The I-SPY findings are reminiscent of the recent subset analysis from Taylor-Retz, the study of the 21-gene recurrence score in nearly 10,000 women with hormone receptor positive, HER2 negative breast cancer and intermediate range scores. The study was designed to evaluate chemotherapy benefit for this category, but subset analysis revealed significant worse outcomes in Black compared to white women with these intermediate range scores, suggesting the presence of tumor biology differences that we do not yet know how to characterize. Others are also doing deep dive research into the inflammatory landscape of Black compared to white breast cancer patients. Yao et al. Utilized the Cancer Genome Atlas and the Women's Circle of Hope database, demonstrating distinct tumor microenvironment signatures for the breast tumors of African American patients. Our research group at Weill Cornell has teamed up with Clayton Yates at Tuskegee, and we identified distinct molecular signatures associated specifically with the triple-negative breast cancers of women with quantified germline genetic African ancestry. Our international research team, the International Center for the Study of Breast Cancer Subtypes, which is now headquartered at Weill Cornell Medicine, has looked at variation in triple-negative breast cancer frequency among populations of women with African ancestry in different parts of the world. We have found very high frequencies of triple-negative breast cancer among Ghanaian women, with Ghana located in Western Sub-Saharan Africa, and relatively low frequencies of triple-negative breast cancer in Ethiopian women located on the east coast of Africa. The frequency of triple-negative breast cancer in African American women is intermediate between what we see in Ghanaian women and what we see in white American women. We therefore believe that the link between African ancestry and triple-negative breast cancer is likely related to germline genetic Western Sub-Saharan African ancestry. SEER registry data mirror these patterns. ER-negative breast cancers are not only more common among African American patients, but also among West African-born patients whose breast cancers were treated in the United States. In contrast, frequency of ER-negative breast cancers is low in both white American and East African-born breast cancer patients. We coined the nomenclature oncologic anthropology to explain these patterns. The colonial-era transatlantic slave trade brought Africans from the west coast of the continent across the ocean to serve as slaves, resulting in substantial shared ancestry between contemporary African Americans and contemporary Ghanaians. In contrast, the slave trade from East Africa brought the ancestors of contemporary Ethiopians further eastward to the Mideast and to Asia. The germline genetic link to TNBC is therefore likely associated with Western sub-Saharan African ancestry. We recruited this brilliant geneticist, Dr. Melissa Davis, to serve as the scientific director for our research program. Melissa is a renowned expert in ancestrally defined germline genetics and ancestry informative markers. In particular, she's worked extensively with the Duffy antigen receptor for chemokines, a gene that has a 1 variant that is closely linked to Western sub-Saharan African ancestry, and this variant is widely referred to as the Duffy null allele. In a study conducted by the YAMBR Consortium, the Duffy null allele was found to be causally linked to differences in circulating levels of various chemokines between African American and white American women. Other studies have demonstrated that Duffy null is the cause of benign ethnic neutropenia. Currently, we and others are exploring its role in cancer and organ transplant disparities. We were therefore excited to have Melissa study Duffy null in our robust international breast cancer biorepository. Most genome-wide association studies in breast cancer have evaluated women with European ancestry, and so the Duffy null allele has been overlooked. We evaluated Duffy null compared to other variants that have been correlated with breast cancer risk, as shown in this table. Our genius biostatistician, Dr. Yale Chen, performed the analyses, and Duffy null was found to be the variant most strongly associated with having a triple negative compared to non-triple negative tumor in both the dominant model looking at overall genotype and also in the dosage model looking at allele presence. As shown in this graphic by the brilliant anthropologist Dr. Sarah Tishkoff, ancestry informative markers are not necessarily random genetic patterns. Many represent locally adaptive traits or genetic variants that clustered in particular populations as a consequence of evolutionary selection pressure to survive specific geographic environments, food sources, or infectious diseases. The Duffy null variant is an example of a locally adaptive trait and aligns with our concept of oncologic anthropology. Duffy null is a variant that confers some resistance to malaria, which became endemic in Western sub-Saharan Africa several thousand years ago because the mosquito host of the malaria pathogen thrives in the climate of this region of the continent. Many of the high-altitude areas of East Africa, in contrast, do not support the mosquito life cycle, and so malaria has a different history in different regions of the continent. Malaria is an extremely deadly disease, and so there was tremendous evolutionary selection pressure on the human genome in malaria endemic regions, favoring any genetic variants that conferred resistance. Duffy null is one variant, sickle cell and the thalassemias represent others. Since Duffy null does not have the adult health consequences of the anemias associated with sickle cell and thalassemia, it became nearly fixed over generations and is seen in nearly 100% of contemporary Western sub-Saharan Africans. This variant traveled with the transatlantic slave trade and because of genetic admixture with Europeans is seen in approximately 3/4 of African Americans. The Duffy null variant travels across to different regions of Africa with an anthropologic event called the Bantu migration, and if you map frequency of Duffy null with frequency of TNBC across different regions of Africa, there's a very close match. Presence of the Duffy protein on red blood cells serves as the portal for selected malaria pathogens to enter the erythrocyte and cause disease. Individuals that are Duffy null lack expression of this protein on their red blood cells, thereby conferring some resistance to the disease. We are now learning that absence of the Duffy protein on the red blood cells also compromises some of the ability to balance circulating levels of various chemokines, and this probably explains the YAMBR Consortium findings that I showed you earlier. Melissa's lab is now teasing out the impact of chemokine imbalance on the inflammatory microenvironment of breast tissue so that we can sort out the precise mechanistic link to triple-negative breast cancer. Melissa's work with the Cancer Genome Atlas has demonstrated that Duffy protein expression in breast tumors is associated with racial ethnic identity as well as survival across all phenotypes. I thank you for your time and attention, and I again congratulate the presenters at this session, and I look forward to future collaborative endeavors that will address breast cancer disparities, utilizing all of the cancer healthcare delivery and research tools that we have available.